So recording. Okay, so in last lecture, we discussed quite intensively about the glycolysis. We're going to talk about the later stages for the metabolism to basically utilize glucose or some other macromolecules to generate energies. So here, we're going to basically, quickly touch on the summary of the glycolysis that we talked about. So glycolysis is conversion of one molecule of glucose into two molecules of pyruvate, as you know, because it has two legs, with the net gain of two molecules of ATP, and it actually comprises like two stages, right? As you can recall, the investment stage and payoff stage, where you use two ATP first, and then will gain 4 ATP in total. You're just gaining 2 ATP. Overall equations right here, and in here, as you can see, you get the two molecules of pyruvate. Importantly, you get also two molecules of NADH, which is a reduced form of NAD+. So basically, they took the electrons from the oxidated glucose, and then that will be actually transported further to the other system, where it generates further, more energies later on. And there were 3 points of controls within this glycolysis. The first one is on the step 1, where hexokinase is involved for that reaction. And that hexokinase enzyme is inhibited by its own product. And the second one is the phosphofructokinase, or PFK, that happens on the step 3. That can be inhibited by ATP and citrate, which makes sense, right? If you have excess of ATP as well as citrate is the substrate for the much later stage. So meaning that there's a log jam, or already there are a lot of stuff that has to be processed. So it'll stop there or inhibited by that, but activated by AMP again, indicative of this cell is now running low in terms of its temperature storage of energy. And the third control point was the pyruvate kinase that happens in step 10, inhibited by ATP, similar to the phosphofructokinase, as well as acetyl-CoA, and the activated by AMP and fructose 1,6-bisphosphate. And the committing step that we talked, whether once it passes that step, it cannot go back, was this PFK reaction, which is on step 3 here. And it's a reversible reaction, so essentially unidirectional, as well as there are a couple more of those unidirectional reactions, including hexokinase step, reaction step, and the pyruvate kinase too, which are basically same as these control points. So these control points basically work as a checkpoint, whether this is cells are ready or not. Once they are ready, just move forward, and then we'll never go back. In addition to ATP, the glycolysis again generates this NADH and pyruvate that have to be dealt or the processed with the later steps of metabolism. And under aerobic conditions, these pyruvate molecules that were generated from glycolysis can be processed through the TCA cycle. And we talked about this briefly, right? That happens right after, not right after, but after the glycolysis to further process the small molecules, the pyruvates, in a slightly different form that we're going to talk about during this lecture. A TCA cycle is basically tricarboxylic acid cycle, also known as a citric acid cycle or Krebs cycle. Basically, these are all the same thing based on what you're looking at. It uses a citric acid or citrate, so it's called the citric acid. It uses a tricarboxylic acid too, so that's why it's called that way. And the Krebs is basically who found this cycle. And pyruvate is processed further for the complete oxidation of this molecule, and its resultant would be just carbon dioxide molecules. And also it produces additional NADH, which is again resulting from the oxidation of a pyruvate, but the NAD plus, the coenzyme itself is reduced, right? So it's oxidation reduction or redox reaction, as well as FDH2, another coenzyme that can work as an electron carrier, so it can be reduced too. NADH can be reoxidized. So again, NADH is the reduced form, right? So it can be go back and reoxidized back to NAD plus in respiratory chain, which is an electron transport chain for again generating more energies. So it's all about breaking down the glucose molecules and then generating as much energy as possible in the form of ATP. In anaerobic conditions, the pyruvate can go through the fermentation to become a different molecule. So that's yeast basically makes the ethanol using pyruvate going through this process of fermentation. And also other microorganisms and animal tissues, especially actively working muscles, can process this pyruvate to make the lactic acid or lactate. These are lactate and lactic acids are different molecules, but they're going to be processed in becoming the same molecule and product eventually as a lactic acid, which is again kind of toxic, making the muscles cramp. And in this process of a fermentation, pyruvate is not oxidized, but it's actually reduced, meaning that it gains electrons instead of losing up the electrons. So it's the opposite way that you can think of pyruvate that is processed in the TCA cycle. But the coupled reaction will be reoxidizing the NADH now back to the NAD plus, right? And then it can be recycled basically and be used in the step 6 of glycolysis, which is the GAPDH reaction that we talked in the previous lecture. And this yeast alcohol fermentation or ethanol fermentation happens in two steps. So the first step would be pyruvate here, which is 3-carbon molecule, becomes the acetaldehyde. So it loses this oxygen basically. And then in this process is catalyzed by pyruvate decarboxylase. So decarboxylase meaning that it loses the carboxylic group, right? The functional group. And the step 2 would be using this acetaldehyde and then further process it to ethanol. And this happens by catalyzed by the alcohol dehydrogenase or ADH. And this reaction is coupled with the NADH that is reoxidized to NAD plus. Again, this NAD plus is going to be recycled in the glycolysis on the step 6, as well as some other reactions basically that requires oxidation, because NAD plus can be reduced, meaning that it can work as an oxidizing agent, right? So don't get confused with these terms. One thing that can be reduced can work as an oxidizing reagent. And this process is the basis for beer and wine making, as you know. But also, not just becoming generating ethanol for the fermentation, animal tissues and some other microorganisms can go through these anaerobic conditions to process pyruvate into lactic acid. So it's called the lactic fermentation. And again, examples is the actively moving or contracting muscles. In here, all oxygen molecule is already consumed because of extraneous working. So that means no oxygens are provided for the oxidizing the pyruvate. So instead of doing that, so it cannot go through the TCA cycle for that process where it generates more energies, but instead, this pyruvate is again reduced instead of oxidized to lactate by this enzyme, lactate dehydrogenase or LDH. And here's basically the reaction. And this process is also known as a step 11 of glycolysis. And as you can recall, I mean, glycolysis comprises 10 steps, right? So it's can be considered as a happening on the next step, which is coupled to reoxidation of NADH to NAD plus. So it's the same thing as the alcohol or ethanol fermentation, which end product, the NAD plus, can be recycled on the glycolysis. And again, muscle cramps is due to this buildup of lactic acid because there's so much of acidity. So it leads to the acidification of the muscle tissues. So it has to be cleared, and it takes a long time, basically. But it eventually will resolve by the clearing, slowly clearing by the bloodstream. So this lactic acid is removed by blood, basically, and that flows into the liver. And once it gets to the liver, then it can be resynthesized into glucose. So it goes back to the original molecule where it has 6 carbons, right? Via gluconeogenesis, which is the process of generating glucose from the smaller molecules. And here's the overall anaerobic glycolysis reaction in muscle. And here's a comparison between the lactate and lactic acid molecule structures. All right, so far we just quickly talk about the 3 different fates of the pyruvate, that it can go through this TCA cycle to generate more energies and eventually oxidize to carbon dioxide in the presence of enough oxygen. Or it can go through this fermentation. It can be lactic fermentation to generate lactate, or the alcoholic or ethanol fermentation to produce ethanol and carbon dioxide. Any questions so far? So now we're going to get to the first fate of those 3, which is really critical. So this is critical because this is basically a central pathway to generate a lot of energies, like in the form of ATP. So it can actually utilize the different kinds of macromolecules, but we're going to focus on glucose first, or glycolysis pathway first for this. But later on, we're going to quickly touch on the other molecules too. But whatever molecules you have to just feed those molecules into TCA cycle to generate lots of ATPs, or again, energy. So here's the TCA cycle. And here we talked about glucogen or glucose that you can get that gets into go through the glycolysis to get the pyruvate. And then there's another process involved to make pyruvate to acetyl-CoA. And once you get the acetyl-CoA, that gets into this citric acid cycle or TCA cycle to oxidize further of a pyruvate. Again, TCA cycle has different names, so you can call it, one of them. All of them are widely used, basically. And it allows basically much more complete oxidation of glucose, so generate much more ATP from here compared to just the glycolysis alone, where you can get only 2 molecules of ATP. Here's the comparison basically for energy. And it has to happen under aerobic conditions. And this pyruvate will be converted into acetyl coenzyme A or acetyl-CoA again. This is the starting point for this TCA cycle. And then ultimately oxidize the carbon dioxide as I just explained. And this TCA cycle takes place in mitochondria, which is one of a huge organelle in the cell. And probably many of you are already familiar with this organelle, mitochondria, but it can be considered as a factory, an energy factory for the cell. So within this mitochondria, there's an electron that is transferred from glucose to NAD plus during glycolysis, as well as other cycles, including TCA cycle. And also FAD is used especially for this TCA cycle. So that's an oxidation. And again, reduction of these coenzymes or the electron carriers. And then ultimately they're gonna be used to reduce molecular oxygens in the much later stages of this metabolic pathway in mitochondria. Again, it's all about generating more energies. And that happens through the electron transport chain that exists in the mitochondria. Excuse me. And in here, protons are pumped across. So let me just step back and quickly explain, the structure of this mitochondria first. So here's the mitochondria. Looks like a little like pin, but it is much more extended structure. Sometimes it has multiple fragments in the cell. Sometimes it's all mingled as just one huge entity. So it's pretty dynamic, but it basically has the same structure as this kind of containing two membranes, each of them called outer membrane and inner membrane. And there's, of course, a space between these membranes, which is called the inner membrane space. And inside of the inner membrane region is called matrix. And there are lots of proteins exist in this matrix as well as in the inner membrane that pass on to the electrons and working for the oxidation. Again, it's just generating a lot of energies. So how it works is that the protons, like any positive charged molecules, including hydrogen cations, are pumped across the inner membrane, mitochondrial membrane, into the inner space right here. So it gets actually out of this matrix. Okay? And this process sets the gradient, and it is both electrical gradient because it's positively charged outside the inner membrane, and also pH gradient too, because the protons are pumped out. So protons like hydrogen cations, so it can change the pH level there. And energy contained in this gradient, basically, the setup, is used to make the ATP. So again, we're gonna talk about much later where the ATP synthesizing enzymes are utilizing this gradient to build for the phosphorylation of ATP molecules to make it into ATP. So here's an example. ATP synthase is the one that is located in the inner mitochondrial membrane, and this process is called oxidative phosphorylation. So oxidative, meaning that it takes up the electrons from a substrate, right? Or adding the oxygen molecules. And phosphorylation is basically adding a phosphoryl group to ADP. So it's the process of making ATP by adding a phosphoryl group by taking up the electrons from a substrate and just put it onto the ADP molecule, or just utilizing. Let me just. Okay. And this process of ATP synthesis is coupled with electron transport chain. Again, it's about using the electron carriers like NAD plus or FAD to moving the electrons from the substrate to the final product like oxygen. And complete cycle of oxidative phosphorylation can result as much as 32 molecules of ATP per glucose. So compared to the just the glycolysis, where you can get 2 molecules of ATP, this is much higher, like a 16 times higher. Basically, there are a lot of molecules that you can get through this process. So that's why TCA cycle is so important. So here are the important and interesting features of TCA cycle, where TCA cycle requires aerobic conditions again, because it's an oxidative phosphorylation process. And TCA cycle is amphibolic, meaning that it is involved in both catabolic and anabolic processes. So it definitely breaks down the acetyl-CoA into the smaller pieces, eventually to carbon dioxide. But also, it provides several intermediates and precursors that will be used for biosynthetic reactions too. so involved in, again, anabolic process. And TCA cycle, as its name refers, is a cyclic pathway. So it regenerates its starting intermediate, which is oxaloacetate that we're going to talk about, but keep using the same molecules again and again. So it's a pretty much like a highly efficient system where it does not actually throw out anything there. And TCA cycle oxidizes acyl groups from the acetyl-CoA basically, and make it into 2 molecules of carbon dioxide. And also it generates 3 molecules of NADH, 1 FADH2, and the 1 molecule of GTP. And this GTP can be converted to ATP, so it's energetically equivalent to that. And again, these NADH and FDH2 will be used later on in the electron transfer chain. So overall reaction is right here for the TCA cycle, where you basically get the acetyl-CoA here, and make it into the 2 molecules of carbon dioxide, and the bunch of coenzymes or electron carriers here. And this TCA cycle is also a pathway for complete oxidation of amino acids and fatty acids too. So not only it's coming from the pyruvate that came from the glycolysis, it can take the amino acids or fatty acids and make it into the acetyl-CoA, as long as it contains a acetyl group, which is like CH3CO group. Then it can provide it to that acetyl group to coenzyme for going through feeding to this TCA cycle. All right. So here we're going to talk about the transition from the glycolysis, where you can get 2 molecules of a pyruvate, to the TCA cycle, where the starting material is acetyl CoA or coenzyme A. So there should be the molecule pyruvate has to be processed and change it to the acetyl CoA. Right? So that transition is catalyzed by this enzyme complex called the pyruvate dehydrogenase, or PDH complex. And we're going to look at its regulation, which is highly conserved and is important as the preparing this TCA cycle. So here's the overall reaction where you put the pyruvate that becomes the acetyl-CoA, and also generates the 1 molecule of carbon dioxide as well as NADH2. So there's a oxidation that is involved for pyruvate, and the role of this transition between the glycolysis and TCA cycle is to simply prepare the pyruvate to the acetyl-CoA to fit into the TCA cycle, and that actually requires an import of pyruvate into mitochondria, because glycolysis, as we talked last week, happens inside the cytoplasm, because all the enzymes as well as the substrates are soluble, right? So it includes the pyruvate too. So pyruvate should be in cytoplasm, but the TCA cycle happens inside the mitochondria. So pyruvate has to be imported into mitochondria. So this pyruvate enters the mitochondria using this pyruvate hydrogen cation import system. And this PDH, or pyruvate dehydrogenase complex, is found in mitochondria of eukaryotes, but as well as some prokaryotes too. So in some bacteria like the E. coli too. And the structure of function of this E. coli PDH complex are well understood, and it's highly conserved for its mechanism as well as some key structures and motifs too. So they can be actually used for understanding better of the other mammalian system like human, which contain large complex, comprises 60 subunits in total. So let's take a look at the PDH structure more closely here. So it has 3 kinds of enzymes, and it's looking like this kind of spiky blob like that, basically. So this PDH complex contains here E1, E2, and E3. So that those are the three different kinds of enzymes. E1 is the pyruvate dehydrogenase, and that have the 12 dimers. 12 dimers, so that means 24 subunits in total. And the E2 enzyme is the dihydrolipoyl transacetylase, so it has 24 subunits, and that makes the works as a scaffold for this like spiky blob structure or the cube structure. So it determines the overall shape of the PDH complex and stabilizes that structure too. And the last one, E3 enzyme, is the dihydrolipoyl dehydrogenase, which have 6 dimers, so 12 subunits in total. So if you add up all these subunits, that becomes 60. So it's a pretty huge complex, but it's finely regulated using these coenzymes too. So they require these coenzymes, different coenzymes for different enzymes. So for E1, it uses a thiamine pyrophosphate or TPP. For E2, lipoic acid in the form of lipoamide that we're going to look at its structure more closely a little later on. And the E3 requires FAD as well as magnesium cation. And also, the NAD plus is loosely associated there too for their activation and recycling of the critical coenzymes here. So they make this kind of non-covalently associated multi-enzyme complex. So there's nothing like the disulfide bond or anything that is involved here, but they're holding together, making this structure using only the non-covalent bonding, like van der Waals forces, or some affinities for the charges and so on. And this PDH, again, pyruvate dehydrogenase complex carries out oxidative decarboxylation of a pyruvate and make this pyruvate into acetyl-CoA. Again, oxidative meaning taking up the electrons. Decarboxylation meaning that it's taking up the carboxylate, which is a COO minus, basically the functional group of the pyruvate. So now let's look at the PDH complex mechanisms for the processing pyruvate to acetyl coenzyme A or acetyl-CoA step by step. So it has 5 steps from 1 to 5 here. And we're going to look at it one by one here. The first step is where E1, again, pyruvate dehydrogenase, with its cofactor or coenzyme TPP, thiamine pyrophosphate, decarboxylates pyruvate. So as you can see here, there's a carboxylic group here, and that is attached and discarded as the carbon dioxide. And then it forms the covalent bonding with the rest of this, which is a acetyl group, to the TPP, which is a cofactor or coenzyme, making the hydroxyethyl TPP or HETPP. And then in step 2, E2 enzyme is used. So it's right here, dihydrolipoyl transacetylase. So it, as in its name says, it uses this lipoamide and then or hydrolipoyl, and then it transfers the acetyl group, right? So this one, E2, oxidizes HETPP that is generated here, and meaning that again takes up the electron there to make the acetyl dihydrolipoamide, which when you look at its structure, E2 contains this lipoamide, which contains the disulfide bond, and this bond is broken, so meaning that it gets the electrons there, so it's going to be reduced, right? And it's oxidizing its substrate, which is HETPP, and then it makes one bond here, diester bond with this acetyl group that used to be attached to this TPP. The rest of this sulfur is going to be again remain reduced. So it's going to be bound to this hydrogen atom here. And this process regenerates TPP from HETPP. So this TPP can be recycled, basically, used for this step 1 again. Step 3 is happening right here, where this acetyl dihydrolipoamide is used and react with the coenzyme to generate acetyl-CoA. So this E2, again, the same enzyme catalyzes the transfer of this acetyl group from the lipoamide to CoA to form that molecule. And this acetyl-CoA, you're going to end up having this acetyl-CoA, which is ready for the TCA cycle. And also will have reduced lipoamide here on the enzyme too. So here's a reduced form of lipoamide, but this cannot be used for this step 2, right? Because this one has to be reoxidized to become making the disulfide bond to be involved in transferring acetyl group from TPP or HETPP. So again, this acetyl group can be just simply gets into the TCA cycle, but this one, the E2 lipoamide oxidized form or SS form, have to be regenerated, recovered. So that happens on the step 4, where the enzyme E3 enzyme here, which is dihydrolipoyl dehydrogenase, works with this FAD prosthetic group that contains also 2 sulfhydryl functional groups, reoxidizes in this E2 lipoamide. So you can just simply think that it's exchanging the bondings. So E3 has this disulfide bond, but the E2 has these 2 sulfhydryl groups that have to have this disulfide bond. So it's giving up this bond here by reoxidizing this lipoamide. But since there's an oxidation, this molecule, the FAD, should be reduced, right? So it's going to be reduced to have these 2 sulfhydryl groups. So again, it's just a simple exchanging of these disulfide bond. And then eventually this E2 goes back to its original form, so it can be again reused for this step 2. But again, E3 is left reduced now, so it have to go back to its original form, here, where it has the disulfide bond, so have to be reoxidized, which happens in step 5. So in step 5 here, the reduced sulfhydryls on E3, which you can see here, are reoxidized, meaning that they're gaining the electrons to become, losing the electrons, to go back to its original form here. But this reaction is coupled to the NAD plus reduction to becoming the NADH and another hydrogen cation here. So it basically takes up these 2 hydrogen atoms to get it reoxidized back to its form. Okay. So that's what I just explained here. And we said that the arsenic is poisonous because it can, basically bound to the magnesium cation in some important enzymes to block its active site or interacting with some polar charged or negatively charged substrate, as well as it can form the pretty stable binding with the phosphoryl group too, which can inhibit the phosphorylation. But also this arsenic is poisonous because it can inactivate directly this lipoamide containing enzymes, such as this E2, or the alpha ketoglutarate dehydrogenase that is used in the TCA cycle. And that basically brings decouples this oxidation or the respiration to ATP synthesis, and then will make the respiration to halt. So even if you feed the glucose and then oxidation happens, you're not going to be able to generate ATP. All right, any questions so far? Yes. Mm-hmm. You mean the each of these processes, or? So it's about the FAD and NAD plus. I'm not sure what you're asking exactly, but let's just follow for this one, right? Oh no, this one. So that means here. So we're right on this fifth one. So what we have now is this two sulfur hydro groups on your E3, and these are reduced form, right? So to recycle this prosthetic group or this enzyme itself, it has to go back to its original form, which is an oxidized form. Does it make sense? So this molecule has to be re-oxidized. So this one has to be oxidized, meaning that something else that is coupled with it for its reaction should be reduced, right? So this one, NAD plus, have to be reduced. I see what you're saying now. So here, FAD is therefore reduced into yeah, I actually skipped this explanation because it can make things a little bit complicated, but it is a really good question. Where this process actually happens in a two-step. So the first step would be this FAD, which is highly favorable to accept the electrons to become the FADH2, will take these two hydrogen atoms or the anions basically to this FAD to make FADH2, and then it'll lead to the disulfide bond of SDs between these two sulfurs. So it's going to be FADH2 and 2 like S here, and then later on this two hydrogen atoms should be removed to truly get the reduced form, oxidized form of this FAD, right? So that's where this NAD plus plays, that reduces the FADH2 to make it into FAD. So I actually skipped this explanation, but it's a good catch, actually. Does that make sense to everybody? So it's a kind of two-step, process. Okay. So here are the advantages of organizing this PDH complex, again, pyruvate dehydrogenase complex enzymes into a single complex. We talked about the making an enzyme complexes and how they can be organized, like totally soluble, or making into some kind of complex, or making into some kind of conveyor belt, or the bound to the membrane and so on. There are different organization methods, right? Especially this PDH complex takes a strategy where it makes a blob, like a complex there, inside a mitochondria in the matrix. And that is advantageous because all enzymes are basically stay very close. And that means their active sites are very close too. And they're handing on and handing off their substrate or intermediates, right? So that means they're going to have the much more efficient way to pass on to these intermediates from first step to the next step, or one to the next and the other enzymes. So it creates this substrate channeling. And here, it limits the diffusion of those intermediates or the substrates out of other places other than just to try to keep it in the complex. So where if those molecules are just diffused out and happen to just collide with the other molecules or enzymes, it can be processed in a different way, resulting in some kind of side product, right? So it limits that. So the rate of the reaction is enhanced. And also, there are going to be little to no chances for side reactions that could occur. And also, it allows the coordinated control of these three enzymes in a different ways, in a more efficient ways. Okay, so, but this PDH complex reaction need to be carefully controlled because its process of making pyruvate to the acetyl-CoA is virtually irreversible because its delta G naught value, the free energy, is highly negative, as negative 33.5 kilojoule per mole. So it can be spontaneously happen pretty easily. So it's energetically favorable, meaning that whenever there is pyruvate, it'll try to just process it further. So there gotta be a way to control that flow. And besides, there's no other way in the mammals to make this acetyl-CoA from pyruvate. So this is the only channel to generate acetyl-CoA for the TCA cycle. So again, have to be carefully regulated. And also, this making acetyl-CoA commits the pyruvate to go through the TCA cycle, or instead of going through the TCA cycle, it can take the other route where it can synthesize the fatty acid too. So it's a critical moment where you have to determine that you have enough energy to store your excessive energy source into the form of fat, or you can just process it further to generate more energies in case you're running low on your energies like ATP. Okay, so how this PDH complex is regulated. So there are largely two different regulations here: inhibition, product inhibition, and reversible covalent modification. And product inhibition happens by the allosteric regulation. So NADH and acetyl-CoA can inhibit the PDH complex. So these two are the direct product of a PDH complex, right? And they can allosterically inhibit the PDH complex, which makes sense because it basically works as a negative feedback loop. So NADH competes with the NAD plus on E3 on the PDH complex, and acetyl-CoA competes with just a coenzyme A on E2 on this complex respectively. And this E2 enzyme stays acetylated, can be stayed acetylated, meaning that there not much of acetyl-CoA that is generated, right? So it cannot accept, in which case it cannot accept the acetyl group from this HETPP, which has the acetyl group on this coenzyme or the cofactor TPP on E1. So again, since you can't process further of this acetyl group transfer to the coenzyme A, it's gonna just stay there. So E1 is stuck basically in that stage. And the second regulation is by reversible covalent modification, which is a phosphorylation of E1 on the specific serine residue to inactivate the enzyme. But that occurs only in eukaryotes, but not in the bacterial PDH complex. We just talked about the structure of a bacterial PDH complex as well as its mechanism. So that although that mechanism is conserved in the mammalian system, this kind of modification-based regulation doesn't happen in bacteria. And this one is accomplished by an enzyme called PDH kinase. Again, for the first phosphorylation, there are two types of enzymes that are involved there: kinase or phosphatase. And kinase is basically ones that adds the phosphorus group to your substrate. And the phosphatase is the other way around, basically taking off the phosphorus group of the substrate. So PDH kinase inactivates E1. So that means phosphorylated E1 will be in an inactive form. And it uses ATP as a donor for the phosphate group. So it actually consumes ATP, uses energy for this inactivation. And that makes sense, right? Because if you have excess ATP, now you want to just stop or at least kind of slow down this process of a PDH complex where it generates more of acetyl CoA. So ATP feeds back and says that you have enough stuff, so you want to just slow down. And as opposed to that, PDH phosphatase removes the phosphorus group again, reactivates the enzyme. So it's going to be a balance between the activity of this PDH kinase and phosphatase to regulate the PDH complex activity. And NADH and acetyl-CoA can activate PDH kinase too. And again, NADH and acetyl-CoA are the products of this PDH complex, right? So there are a lot of them there already. So you want to slow down. In which case, these products, can activate the PDH kinase, which will inactivate the E1 of the PDH complex. So it works in the exact same way as the product inhibition in terms of their direction, and pyruvate and ADP instead inhibits the PDH kinase activity. So works in the other way, because you have a lot of pyruvate, so it has to be processed, or there's a lot of ADP instead of ATP, so you need to generate more energy there, in which case kinase is inhibited by that, and then will activate the E1 enzyme activity. Okay. Yes, question. Your question was that pyruvate? So pyruvate will inhibit the kinase, meaning that the kinase will not be as active, right? So it will not efficiently inactivate E1. So if it doesn't inactivate E1 too well, then it's going to be actually activating the E1 activity, which is one of a PhD complex. So in the end, you're going to get the more activity for PDH complex, which will lead to generating more, processing more pyruvate into the acetyl-CoA. So it's kind of multi-step, but again, working in the kind of same way for all the other regulations, including allosteric regulations. Any other questions? Yep. What? The question was that do we need to memorize all the steps? I would say it would be great, if you can understand and memorize all the steps. Of course, you're going to be able to answer all the questions, right? I wouldn't ask the particular kind of player on the particular step that is required, but I'd hope you to have the overall kind of picture of how the mechanism actually works, what kind of steps are involved there, what kind of redox reactions there are utilized, things like that. It's more like the mechanisms and concepts rather than the facts that I'll be asking. That doesn't mean that I'm not going to ask anything about the facts. I could ask, what is required for as a cofactor for E2, which is right here, right? Repork acid, something like that. Definitely can ask that. Okay. All right, so now the pyruvate is processed to acetyl-CoA. So we just finished the glycolysis and the transition from pyruvate to acetyl-CoA. Now it's the beginning of TCA cycle. So we're going to talk about this TCA cycle, which involves 8 steps, as you can see here. And we're going to talk about the mechanisms one by one. So I know that there are a lot of pathways and mechanisms and transitions are involved there, but they are really important because many of these enzymes are used in other biochemical processes too. So and using the same mechanisms, so they are really important. Again, and this TCA cycle is a central pathway of a metabolism. So we must actually understand all the steps. So the step 1, which is on top of this TCA cycle, is getting the acetyl-CoA and making into the citrate. So it's called the citrate synthase reaction, where the enzyme called citrate synthase involved here. And what it does is basically condense down this acetyl-CoA and the oxaloacetate, which is an end product of the TCA cycle. So this is a cyclic pathway, right? So that's going to be the last step, the product of the last step. Oxaloacetate combined together to form a citrate. So as you can see here in the molecular structure, oxaloacetate has 4 carbons, and acetyl-CoA, the acetyl part, has 2 carbons here, as you can see. So they are condensed down or combined to make the C6 6 carbon molecule citrate. And that has involves the hydrolysis of thioester bond of the acetyl-CoA, which is right here. So here's a sulfur, and here's the oxygen. So it forms the thioester bond here. And since this is a high energy bond or high energy molecule, it will release a lot of energy with the delta G naught, negative 31.4 kilojoule per mole. So it's highly negative. So it's highly spontaneous reaction. But this reaction, this reaction is virtually irreversible, meaning that it's gonna energetically favorable, but also because it works so well, you got to have some kind of regulation pathway there to in case it has to be stopped and things like that. So there are regulated process. The regulations are involved there, so it's highly regulated by several methods, and it's also allosterically inhibited by the succinyl-CoA, which is a product of step 5 of a TCA cycle. So basically, it's just it's a intracyclical regulator that tells that there are log jam of this molecule, or you have too much of this, so you gotta just slow down there. And NADH is another one that inhibits allosterically of this citrate synthase, which is a product in the all 3 steps in this TCA cycle. So again, it works as an indicator of energetic status of the cell, where many things is oxidized already. So you gotta just slow down there. The second step is the isomerization of citrate to isocitrate. And when you look at these 2 structures, citrate has the H and OH here, as opposed to isocitrate has a flipped order, where the OH, the hydroxyl, is on top, and the hydrogen atom is on the bottom. And that doesn't just happen simply flipping this order or the bonding here. That involves more like the detaching these 2 functional groups in the form of a water molecule, and then attach a new water molecule in the flipped order here. Question? So again, kind of related question. I mean, these are really important questions, I guess. The question was that do we need to memorize all the molecule structures and their formulas? No, you don't need to. But at least you need to know the critical functional groups and their mechanisms. For example, we're talking about step 2 here. where it makes the isomer, there's isomerization, right? So you should understand where it happens and how it happens. What kind of functional groups are involved here? No, that's important. But you don't need to memorize all these, carboxylic group or the methyl group and things like that, everything here. Of course, I mean, that'll help for you to, easily answer other questions, but not too required. So this reaction involves the intermediate called cis aconitase, which is the form where it's a citrate without this H and OH function, two groups here. And that is catalyzed by aconitase. That's the name of the enzyme. And this isomerizes tertiary alcohol to secondary alcohol. So since the organic chemistry is a prerequisite of this course, we must know what the tertiary alcohols and secondary alcohols are. Can anyone explain what these are? Yes. Very close, actually. By 3 here. There's only one OH group, hydroxyl group, but there are 3 carbons, any kinds of carbon groups. So you can see 3 carbon here, right? There are different groups, but anyway, that is attached to the carbon that has an hydroxyl group. So that's a tertiary alcohol, but it changes to secondary alcohol where the carbon that has the hydroxyl group have only 2 carbons. As you can see, the other one is just hydrogen. And that is important because that makes it easier to oxidize this isocitrate in the next step. And that again happens in the 2 process using this intermediate. And basically what happens here is that the water molecule is abstracted from the citrate, and then another water molecule is added back to this cis aconitase but the opposite positions to make the isocitrate. And its delta G naught value, free energy, is positive value, positive 6.7 kilojoule per mole. So it's supposed not to happen, right? It's not a spontaneous reaction unless there's an energy input there, but it still happens because it is coupled to the other exergonic process, which happens in the next step. So it's a kind of general theme in any kind of biological or biochemical reactions where endergonic reaction is coupled to exergonic reactions to move it kind of forward, pull everything forward. So the next step, which is an isocitrate dehydrogenase reaction, has the negative value for its delta G naught. So they can be simply coupled, which is a step 3 here. Again, I just called it here. And it's called, again, oxidative decarboxylation of isocitrate. So oxidative, meaning that it loses electrons. The substrate loses electrons. And also decarboxylation, so it's gonna lose carboxylic group, carboxylate, which is a COO minus or COOH. So let's take a look at what it happens. But it's a first oxidative reaction, and every oxidation counts, important, because they can hand over the electrons, can generate the reduced form of electron transport or electron carriers. And these reduced electron carriers are used for generating ATP later on. So whenever you see that there's NADH happens or oxidation happens, that is related to new ATP molecule in the end. So this process is catalyzed by isocitrate dehydrogenase. You can see it here. And that's, again, 2-step reaction where the first step is the oxidation of the hydroxyl group at C2 of this isocitrate to make an intermediate called oxalosuccinate. And this the reduction of NAD plus reaction is coupled to this to take over the electrons from this isocitrate. And then the next step is the decarboxylation of oxalosuccinate to produce this alpha ketoglutarate, where it loses here the carboxylate on C2, and since it has COO minus, it becomes to to carbon dioxide. And this intermediate product, oxalosuccinate, is unstable and easily decarboxylated to alpha ketoglutarate with its delta G naught value negative 8.4 kilojoule per mole. So again, this is a spontaneous process. So it can pull the reaction of the step 2, the previous step, forward. And this NADH that is generated from this step enters the electron transport chain later on, still within the mitochondria. So everything is happening in mitochondria, but it enters the other pathway, metabolic pathway, and can be used in the oxidative phosphorylation that leads to the generation, the synthesis of ATP. And this molecule, the enzyme isocitrate dehydrogenase, is also allosterically regulated. So ADP and NAD plus activates this enzyme, as opposed to ATP and NADH inactivates or inhibits this enzyme. And again, it makes sense, right? Because it's all about the ratio. Here, as it says, it's all about the sensing the ratio of NAD plus and NADH and ADP and ATP there, which is which shows the metabolic status of the cell. So if you have the high ratio of these two, meaning that you have a lot of like what is it, the oxidized form of NAD plus or the ADP, that means you have to generate more ATP. You have to process these molecule, the sugar molecule, more. So that's means a green light. So it'll basically activates this pathway. So the next step, step 4, is about the oxidative carboxylation again of the alpha ketoglutarate. So since it has another carboxylate here, this one can be detached and converted to the carbon dioxide here, and will generate the succinyl-CoA with the coenzyme A here. And also here, this reaction is coupled to the reduction of NAD plus that becomes to the NADH. And this reaction is catalyzed by alpha ketoglutarate dehydrogenase. And this alpha ketoglutarate dehydrogenase enzyme is also forms a large multi-subunit enzyme complex that have a similar structure to PDH complex, and that also acts using the same mechanism there. So this complex, the enzyme complex, is composed of these 3 different type of enzymes, which are alpha ketoglutarate dehydrogenase. So it's similar to pyruvate dehydrogenase. And the second one is dihydrolipoyl transsuccinase. So it's similar to the PDH complex, the second one, which is acetylase. And the third one is the same as the third E3 in the PDH complex, which is dihydrolipoyl dehydrogenase. And again, they use the same mechanism that goes through the step 1 to step 5 that we just talked before to making the pyruvate to acetyl-CoA. But instead, it uses alpha ketoglutarate to generate succinyl-CoA. And it requires similar cofactors or coenzymes too, like magnesium cation or TPP, lipoic acid, and FAD. So exact the same coenzymes. And it also produces carbon dioxide, diester product, and NADH2. So again, using the same, exact same pathway with the delta G naught value negative 33.5. So it's pretty much identical too. And this enzyme complex is also regulated by positively by AMP and negatively by NADH and succinyl-CoA. Again, it's about its product. It's detecting its product itself and can work as an inhibitor for this enzyme complex. So the step 5 is the conversion of this succinyl-CoA to succinate with the production of GTP. And this is the only step where substrate level phosphorylation is involved in TCA cycle. And this substrate level phosphorylation is meaning that a substrate provides the energy for the phosphorylation of GTP or ATP. And also means that it's not linked to the electron transport chain, which happens much later in the TCA cycle. And this succinyl-CoA has the high energy intermediate because it has this thioester bond here. So it contains a lot of energy there. So upon the breakage of this bonding, it can release a lot of energy to drive this reaction forward. And it provides the energy to generate GTP2, which is a high energy phosphate. And just already talked about this, but the hydrolysis of diester bond, which happens on this breakage of the succinyl-CoA, has this releases 33.4 kilojoule per mole, and that has the enough energy for synthesizing GTP, which requires 30.5 kilojoule per mole. And the net delta G naught value would be still negative. So it's going to be a spontaneous or the energetically favorable reaction. And this GTP exchanges the terminal phosphoryl group that is attached during this process will get to the ADP to produce ATP and GDP. And that process where the GTP hands over the phosphoryl group to make the ADP to ATP is catalyzed by nucleoside diphosphate kinase. So far we just talked about the 5 steps from the first step to 5th step of the TCA cycle, and that is starting from the acetyl-CoA that was generated from the transition of the pyruvate to acetyl-CoA by PDH complex, and that goes through all the intermediates and generate the other products to become succinate in step 5. And that is eventually it started with this acetyl-CoA and oxaloacetate to condense it down to citrate and liberates the 2 molecules of carbon dioxide and also generates 2 molecules of NADH, which is one's here, the other's here, step 3 and 4. And also 1 GTP molecule on step 5, which will be converted to ATP later on, and it ends up with the succinate here. And the remaining steps is a further oxidation to make it into a carbon dioxide. And the complete this cycle to regenerate the starting material, which is oxaloacetate there. All right, so we're going to talk about the step 6, 7, and 8 here. 6, 7 is the oxidation of succinate to fumarate with the reduction of this FAD, another electron carrier. And that reaction is catalyzed by succinate dehydrogenase. And this is the only enzyme in the TCA cycle that is membrane bound, especially on the inner mitochondrial membrane. And as I just said, all the other enzymes as well as the substrates are in the matrix. But this is the succinate dehydrogenase is only the only enzyme that is bound to membrane. That will change the dynamics here of the molecule processing here. And this succinate dehydrogenase is also part of electron transport chain, which happens on the inner membrane of the mitochondria. So this is basically just a one enzyme that can be used for both pathways, but separately. They're not linked together directly, but using, basically doing the same thing here. And it reduces the FAD to FADH2. So it takes up the two hydrogen anion, right, hydrides, meaning that two electrons, just like the NADH. But this is the not sufficiently exergonic process because the I'm sorry. So here's the energy that can be generated, that can be used, basically. What is it? It's an exergonic process where it can provide the energy to other coupled reaction here. And that coupled reaction could have been a reduction of NAD plus, just like the other processes, but it's not. Instead, it uses FAD because it cannot produce or provide the sufficient energy to reduce NAD plus, which requires more energy for its reduction. But it's still enough energy to reduce FAD molecules, so FAD is used instead. And that is telling that FAD requires less energy for its reduction, right? And that is because FAD is a stronger oxidizing agent. So it's easier for this FAD to accept electrons to become FADH2 instead of NAD plus is reduced. So, but how come, FAD isn't used for all the processes? Because it requires less energy for reduction. So it can essentially, achieve the oxidation because it can work as an oxidizing agent for any kinds of molecules, right? So instead of using NAD plus, all the pathways and all the molecules could have used FAD instead, but why not? Can anyone think of any possible regions that the cellular system uses FAD, NAD plus instead of FAD in most processes? Any ideas? Because FAD seems to be a better one, That's a possibility too, yeah. There's no right or wrong answers. I just want you to just think about this. It could be the regulation issue, but eventually it'll be about the that is what? So because it is more stable. Both of them are pretty stable. But eventually, it requires less energy for the reduction. So oxidizing will require less energy too. So maybe you can consider that as a little bit less stable. But eventually the reason would be because NADH can generate more energy, more ATP rather than FAD. So it's more efficient in generating energy in the electron transport system or the electron transport chain. So here's a step 7 where the fumarate is hydrated, meaning that it takes up the water molecule here to generate the L-malate. So it happens in the stereospecific manner. So instead of generating like L or D malate there, like the racemic mixtures or so, it always makes the L malate there. And that's because of this enzyme fumarase that prefers making this L-malate. And this process involves a transaddition of elements of water molecule, because the one here, it can be, the water molecule is broken to hydrogen and hydroxide, and then these will bound to these different sides of this carbon, breaking the double bonding between the carbon atoms here. And this reaction is catalyzed by fumarase. And again, it has a specificity for L-malate, as you can see, where the hydroxyl is on your left side, so it's an L form. And the last step, the step 8, is the oxidation of this L-malate to going back to the oxaloacetate. And also the reduction of NAD plus to NADH is coupled to this reaction. This reaction is catalyzed by malate dehydrogenase. And it is a highly endergonic reaction where delta G naught value is 29.7 kilojoule per mole. So it's really a positive value. So it will not actually happen at least at the standard state. When you think about this equation for the delta, making the relationship between delta G and KEQ, since this delta G naught is a positive value, this RT, ln KEQ must be negative, meaning that RT are positive values, so ln KEQ must be negative. And also that means instead of the natural log, the KEQ should be smaller than 1. And KEQ by definition is the ratio of concentration of the products and the concentration of reactants. And that is smaller than 1, meaning that you're going to have much less products compared to reactants. So the product, which is oxaloacetate, must be kept in low concentration, very low in the mitochondria at equilibrium. But this reaction still goes forward because this oxaloacetate, which is a product here, can be kept low. And it can be achieved by the immediate clearing of this oxaloacetate by the reaction in the step 1, which is highly exergonic with the delta G naught value negative 31.4. So it's again the coupled exergonic and endergonic reactions there. So here's a quick summary of TCA cycles as well as PDH complex reactions that we just talked about. So the delta G for the one cycle of TCA is the negative 40 kilojoule per mole. So it's energetically favorable, meaning that it will happen, by itself. And summary of this PDH complex has this basically a reaction right here, where the pyruvate is used to make the acetyl-CoA as well as NAD plus is reduced to NADH by getting the 2 electrons. And here's a TCA cycle reaction that we just talked about. And the overall reaction would be here, where it generates not only carbon dioxide here, but also here NADH, 4 molecules of NADH and FADH2, and the GTP2. All of them are basically the source of energy, and that completes the metabolism of glucose and makes it into the single carbon molecule here. But so that means these all 6 carbon atoms in the glucose is completely oxidized and liberated in the form of carbon dioxide. And this reaction will ended up having 2 carbon dioxide that is released by the PDH complex and 4 carbon dioxide molecules by TCA cycle. And think about it, where overall reaction says that there's only 3 carbon dioxide. But when we're going back to the glycolysis, the one glucose generates two pyruvates, right? So this starting from two pyruvates, meaning that this overall reaction will have twice much of all these molecules. So that includes 6 carbon dioxide in total. And think about the balance, energy balance calculated in the ATP equivalents. I just briefly talked that the NADH and FADH2 eventually gets into the electron transport chain to generate ATP. And this one molecule of NADH is equivalent to 2.5 ATP, as opposed to FADH2, one molecule is equivalent to 1.5 ATP. And that's why NADH is a better electron carrier, because it will generate more energy there. And when you calculate that from the glucose, where it has lots of NADH on the different steps, as well as one molecule of GTP per one cycle. But of course, there's a 2-acetyl-CoA, so that's going to be 2 GTP that converts to the 2 ATP. And if you simply just put this equivalent for the ATPs, then you're going to end up having 32 ATP. So that's a lot of them, right? And this is the last slide where it's all about the oxidation of glucose and breaking into the carbon dioxide, generating a lot of energy there. And this is all about making the ATP, but because there's a lot of energy released from sugar or the glucose, that doesn't mean that everything will be immediately converted to ATP. But they can be stored in the form of reduced coenzymes like still NADH and FADH2, and can be used in the other reactions, or just stay as they are until the energy is required, more energy is required. And here's a complete oxidation of a glucose reaction is written, and that can be written as the 2 half reactions, one for the oxidation of glucose, which is right here, that and another one is a reduction of molecular oxygen, which is right here. And when you combine these two, you're going to end up having this simple glucose oxidation reaction. So that's it. And you guys have the happy Thanksgiving, and we'll see you a week after, I think.